Dear ladies and gentlemen, my name is Ruslan Gazizov, and I'm a senior research worker at the GeoSplit Research Laboratory. Our company is actively engaged in an innovative well marker study without downhole operations for the oil and gas industry. We are in the GeoSplit Laboratory, where I will tell you more about the principles of marker diagnostics technology based on quantum reporter markers. Light is an electromagnetic radiation that falls within a certain portion of the electromagnetic spectrum. The behavior of EMR depends on its wavelength, frequency, and energy. Visible light is usually defined as having a wavelength in the range of 400 to 750 nanometers (nm). We see these waves as the colors of the rainbow, where each color has a different wavelength. Red has the longest wavelength, and violet has the shortest wavelength. When all the waves are seen together at the same time, we see it as a white light. Luminous simply means giving off light. Scientists have many words to describe the different kinds of luminescence: bioluminescence, chemiluminescence, electroluminescence, photoluminescence, fluorescence. Luminescence emission occurs after an appropriate material has absorbed energy from a light source. For example, if a material absorbed blue light, then emitted green light. In general, the emitted fluorescent light has a lower wavelength and lower energy than the absorbed light. This phenomenon is commonly referred to as Stokes shift. In fact, fluorescence occurs in nearly all substances. However, those that have more pronounced properties are called fluorescent dyes. Historically, the first substances that clearly showed fluorescent properties were complex organic compounds, most often extracted from natural sources: Congo red, acrylide, chlorophyll. Fluorescence has many practical applications, such as medicine, chemical sensors, fluorescent labeling, and most commonly, fluorescent lamps. A special group of fluorescent compounds is quantum dots. In our case, we will talk about colloidal quantum dots, which are semiconductor nanocrystals. Why is it called a quantum dot? Quantum dots exhibit properties that are between those of bulk semiconductors and discrete atoms or molecules. In semiconductors, light absorption generally leads to an electron being excited from the valence to the conduction band, leaving behind a hole. The electron in the hole can bind to each other to form an exciton. When this exciton recombines, for example, the electron resumes its ground state, the exciton's energy can be emitted as light. When the size of the exciton becomes comparable with the size of a nanocrystal, the electron energy levels split due to the quantum confinement effect. As a result, in the larger quantum dot, the red, lower energy, is its absorption onset and fluorescent spectrum. Conversely, smaller dots absorb and emit a more blue, higher energy light. So, why are quantum dots better than organic fluorescent dyes? Since traditional fluorescent dyes are complex organic molecules, they can very easily oxidize, destruct, or change in structure. This leads to a reduction of their fluorescent properties. At the same time, quantum dots are more chemically and thermally stable and are resistant to the action of oxidizing agents. For example, LCD displays based on organic fluorescent dyes, OLED, can burn in after long-time use, but LCD displays based on quantum dots, QLED, are much more durable. Another advantage of quantum dots in comparison to fluorescent dyes is the narrow and symmetric emission spectra. In the top picture, green light is the fluorescent emission spectrum of organic dye, and full width at half maximum, FWHM, is about 160 nanometers. The bottom picture shows the fluorescent emission spectrum of quantum dots with a full width at half maximum, FWHM, of about 40 nanometers. Another advantage of QDs is the broad excitation profiles and high extinction coefficient. Due to the broad excitation profile, the quantum dot will emit bright light when excited about almost any type of light, while the fluorescent dye must be excited by light within a certain wavelength range. High-quality quantum dots are well suited for optical encoding applications due to their durable structure, broad excitation profiles, and narrow emission spectra. The use of quantum dots in GeoSplit technology allows to achieve high accuracy and efficiency of diagnostics. Quantum dots of 2 to 10 nanometers in size are placed inside insoluble polymeric microspheres with a size of approximately 1,000 nanometers. Depending on the type of quantum dots inside the microsphere, various signatures of marker reporters can be obtained. The use of 6 types of quantum dots and the large number of possible combinations allows us to create 63 different signatures of marker reporters. This image shows microphotographs of GeoSplit, a marker reporter sample. It can be clearly seen that polymeric microspheres are well-rounded and have a narrow-sized distribution. In the top right corner is the Transmission Electron Microscopy (TEM) image of a synchro microsphere. The small dark particles inside the microsphere are quantum dots. Various types and combinations of fluorescent marker reporters, 1 micron in size, are introduced into the polymer materials. For example, polymer coating of propent. An important microstructural feature of a polymer is its architecture and shape. There are many types of polymer architecture, including linear, cross-linked, branched, and network types. GeoSplit marker reporters consist of a network polymer and therefore have good thermal resistance and chemical stability. This is why marker reporters retain polymeric microspheres during all the stages of GeoSplit technology, which allows it to achieve high accuracy and efficiency of diagnostics. However, polymer coating propent or polymeric powder granulate consists of a practically cross-linked polymer. We can show the basic principles of makers containing polymeric materials in the case of polymeric grains for well filters. The scientific name for this type of plastic is non-homogeneous microfilled composite. Its schematic structure and real microphotography are shown here. It consists of different polymers with different functions: insoluble polymer network as the matrix of the composite, soluble fillers, and GeoSplit marker reporters. The fillers are washed out by the fluid, which carries marker reporters from polymeric material. Depending on the composition of the filling material used, the polymer can be either hydrophilic, dissolves with water, or oleophilic, dissolves with oil. Let's consider hydrophilic or oleophilic properties in detail for the example of propent. The top picture shows a sample of oleophilic propent, and it is clear that oleophilic propent is not moistened with water. In the bottom picture is a sample of hydrophilic propent, where it can be seen that the hydrophilic propent is well moistened with water. The markers from the oleophilic propent are washed out directly into the oil phase, then from the hydrophilic one directly into the water phase. Moreover, marker reporters of 1 micrometer in size can't migrate from the oil phase to the water phase, or vice versa, due to the significant surface tension of water. Surface tension is a phenomenon that allows water striders to float and slide on a water surface. As a result, a selective analysis for each part of the oil fluid can be obtained, which improves the accuracy of the method. To summarize the above, quantum dots, 2 to 5 nanometers in size, are incorporated into polymeric microspheres called GeoSplit marker reporters, 1,000 nanometers in size. These are placed inside GeoSplit products, such as polymeric coated propent. The markers are then washed out by the fluid from polymeric coating. Collected samples of reservoir fluid are sent to the lab for complete analysis. The analytical marker reporter's identification is based on an instrumental method called flow cytofluorometry, or simply cytometry. Flow cytometry is a technique used to detect and measure physical and chemical characteristics of a population of cells or particles. For reference, the linear size of a red blood cell is about 7 micrometers compared to marker reporters that are about 1 micrometer in size. The basic principle of flow cytometry is as follows. A particle suspension is run through a cytometer. Sheath fluid is then used to hydrodynamically focus the particle suspension through a small nozzle. The tiny stream of fluid carries the marker reporters past the laser light 1 particle at a time. A detector in front of the light beam measures forward scatter, FS, and several detectors to the side measure side scatter, SS. Fluorescence detectors measure the fluorescence emitted from GeoSplit marker reporters. Let me remind you that the marker reporters are microspheres incorporated with quantum dots that can fluoresce in different colors. So, each microsphere in the analysis becomes a point in the 15-dimensional coordinate space, but it can still be clearly identified. In the image is a cytometer used in our laboratory, along with examples of 2-dimensional dot plots of data gathered by flow cytometry. Because cytometry is a technique commonly used to detect population of cells, it often uses aqueous and nearly aqueous solutions. The analysis of the samples would be very simple if the fluid from the well was pure water with markers. However, the real fluid is oil, a mixture of oil and water, and emulsions and various impurities including sand, clay, salts, gas bubbles, and organic contaminants. It is a complicated and time-consuming task to separate all impurities without losing a significant number of marker reporters. We solve this problem using a combination of various physical and chemical methods: selective filtration, centrifugation, concentration, and sorption. As a result, we obtain a small vial of pure liquid containing marker reporters. We could spend a significant amount of time analyzing the data obtained by the flow cytometer using the manual method. Let me remind you that each marker reporter in the analysis becomes a point in the 15-dimensional coordinate space. Moreover, the analysis becomes long and complex when the data contains a large number of signals and signatures, so the chance of human error is significant. To analyze flow cytometry data, GeoSplit uses an innovative approach based on artificial intelligence, or machine learning. The software package developed by our team is based on a machine learning method called random forest. The artificial neural network is trained on referee, samples of clean marker reporters or propens, and the so-called decision tree is built with each tree having a certain depth. At every depth stage, particles are sorted according to certain characteristics, for example, if the particle emits green light or not. As a result, while passing through such a tree, the marker reporters of the unique code fall into a strictly defined basket. Then, the random forest builds multiple decision trees and merges them together to obtain a more accurate and stable prediction. Then, a mixture of a large number of marker reporters is examined on the created trees. The neural network recognizes marker reporters of each signature and determines the consistency of the initial mixture. In general, the innovative method of analysis used in GeoSplit allows it to process a large data package with a tunable accuracy in a short time period. This also eliminates the possibility of human error. The markers are then washed out by the fluid from polymeric coating. Collected samples of reservoir fluid are sent to the lab, where the separation and analysis proceed. Further on, the flow cytometer data process uses an innovative approach based on machine learning. As the result, we obtain well inflow profiles. Herein lies the benefit of the complicated GeoSplit technology. The materials based on the use of marker reporters created from quantum dots can be used for a variety of solutions, such as horizontal wells stimulated with multi-stage hydraulic fracturing, horizontal wells without fracturing, vertical wells stimulated with hydraulic fracturing, and well integrity solutions. All of these solutions for production logging were created within GeoSplit in close cooperation with oil and oil field service companies. When the industry has a need for new solutions, we are always ready to collaborate to solve the problem through an innovative solution that provides valuable information. As Rothschild said, "He who owns the information owns the world." Thanks for your attention.